Professor Kell, your lecture is intriguingly titled Axe Soup, Why P-Value Can Be Greater Than 5%. Can you explain this axe soup metaphor to us?
Of course. Axe soup comes from a folk tale in which a tasty soup is made starting with an axe, clearly not edible, and then adding other ingredients like onion, potatoes, bay leaf, and salt. Alone, most of these ingredients aren't appetizing, like the salt. The axe isn't edible at all. But together, they create a satisfying dish. In science, particularly in gene networks, the same principle applies. Individual components may seem insignificant, but their combination produces meaningful outcomes.
So, you're suggesting that in biology, something seemingly insignificant can still be essential?
Exactly. When we study genes, we often use statistical tools, like the p-value, to decide if a gene is significant. Genes with high p-values are usually ignored. But like the salt in the soup, some statistically insignificant genes can play central roles when you consider the entire network.
Could you give an example from your own research?
Certainly. We studied resistance to 5-fluorouracil, 5-FU, a long-standing chemotherapy drug. Working with Professor Raman Zubarev at the Karolinska Institute, we developed cancer cell lines resistant to 5-FU. We compared their protein expression and activity to sensitive cells using both expression proteomics and a functional method called PISA proteomics. One gene, SOD1, barely changed in expression. Its p-value suggested no importance. But our network analysis showed it was a master regulator in resistance.
That's fascinating. How did you uncover SOD1's role if its expression data didn't reveal it?
We integrated multiple data types, transcriptomic, proteomic, and activity profiles, and mapped regulatory networks. Our algorithms identified positive feedback loops that sustain drug-resistant growth. SOD1 sat at the center of these loops. Its regulation happens mostly at the protein modification level, not at transcription, so expression data alone misses it.
And how does this discovery translate into therapy?
Our analysis highlighted uracil, a low-toxicity molecule, as a top candidate for combination with 5-FU to reverse resistance. Historically, uracil and its currently used derivative eniluracil were known to enhance 5-FU, but the role of uracil had been largely forgotten. We confirmed experimentally that uracil restored apoptosis in resistant cells.
So the takeaway is that the salt in your soup, SOD1, was essential, even if statistics didn't say so?
Precisely. In complex systems, the key player may not be the one with the lowest p-value. We must look beyond isolated statistics to the network context. Like in the soup, the flavor comes from the combination.
A great reminder that in both cooking and science, it's about the recipe, not just the individual ingredients. Thank you, Professor Kell.
Thank you, Alice, and see you next time.